a warm welcome everybody to today's Immunoprecipitation Workshop all about IP. My name is Astrid, and I would like to start with a brief introduction about myself and the company, and then we will go through all the slides, and in the end we will have a Q&A session, and please feel free to post all your questions already in the chat. So welcome again. My name is Astrid, and I did a PhD in biochemistry. Then afterwards, I worked in tech support for a lab device company. And since 2019, I'm part of Chromotech and the Protein Tech family. I'm a product manager, and one of my main tasks as a product manager is also to do tech support. So I know all about IP, and I would like to share my knowledge today with you. I would like to start also with a brief company introduction. So Protein Tech is a leading manufacturer of antibodies, immunoassays, and proteins. And we have antibodies against more than 13,000 targets. We also offer ELISA kits, IHC kits, and cell separation kits. And since 2020, Chromotech is also part of the Protein Tech family. At Chromotech, we develop nanobody-based reagents for immunoprecipitation, for immunofluorescence, and also for protein purification. And the third product line that also belongs to the Protein Tech family is Humankind. Under the Humankind brand, we manufacture cytokines and growth factors, and all our bioactive proteins are expressed in human cells. They have a native folding, native post-translational modifications and glycosylations. And we offer research use only grade, but also GMP grade. And what makes Protein Tech so special is, compared to other antibody companies, that we ensure that our products are only sold under the Protein Tech name and only are sold by us. So you won't find our products under a different label. And also what's important for us is full transparency. So we try to share everything with the customer about the product performance and the QC, and product validation is also one important part of the company. And with this, I would like to start the workshop all about IP. Here you see all the questions that we will answer during today's workshop. We will start with what is immunoprecipitation, how you could select the right antibody and metrics for your immunoprecipitation, when you should use a nanobody or an antibody for IP, how you can optimize the different steps of an IP. We will also talk about necessary controls, and in the end, we will have troubleshooting sessions where we go to together through different troubleshooting cases. And so with this, I would like to start. What is immunoprecipitation? Immunoprecipitation, or short IP, is a technique to isolate a protein of interest. We will always use in the following slides the abbreviation POI. And the POI is isolated out of a protein extract using an antibody that specifically binds to the protein of interest. You see it here on the example on the left side. So you start with the antibody that is coupled to a matrix, and then the antibody is incubated with the cell lysate. The protein of interest and potential interaction partners are enriched on the matrix and bound by the antibody. Then unbound proteins and lipids are washed away. And in the end, you only have your antibody that binds the protein of interest, and then you elute your protein of interest, and that's the last step of the IP reaction. So before you do an IP, you have to think about which antibody is right, which tool is right for your immunoprecipitation. And when you select the antibody, there are a couple of do's and don'ts that may make sense to follow. So we recommend that you use an antibody that is validated and that is especially validated for immunoprecipitation, because not every antibody that works in Western blot also works in IP. Western blot antibodies often detect the denatured epitope, and for an immunoprecipitation, you usually need an antibody that can detect the native epitope of your target. When you can select between different antibodies, we recommend that you have an antibody that has a high affinity to the target and also a high purity. And monoclonal antibodies are usually preferred over polyclonals because they show less unspecific binding to other cellular proteins. Depending on how you design your assay, you can either use your protein of interest without a tag, or in some cases, you can overexpress your protein of interest with a tag. For example, GFP and flag tag are very common tags that are used in IP. And when it comes to the metrics, you can also decide between different beads properties. There are solid beads, and there are also porous beads, and some beads are separated from the supernatant by centrifugation, and for others, you use a magnet. And after you have chosen your antibody, then you can think about how you immobilize the antibody on the matrix, so on the bead. And the easiest way is to use just protein A and G beads. There you just incubate your antibody with the beads, and it's a non-covalent interaction between the antibody and protein A and G beads that is formed. If you would like to have a more tighter interaction between the antibody and the beads, you can also cross-link the antibody. There you have an irreversible cross-linking to protein A and G that is located on the beads. Or as a third option, you can also covalently bind the antibody to the beads. So you can bind the antibody directly without the use of protein A and G. The cross-linking and the covalent interaction are usually suitable when you want to have do your IP under very harsh conditions. So for example, when you use a very harsh washing buffer, you would like to ensure that your antibody is not released from the beads under the harsh conditions. However, when you do covalent interaction of the antibody or cross-linking, you should keep in mind that this can also modify the paratope of the antibody, and the antibody may not bind then the epitope anymore. That's the reason why most researchers go with the first option, so just use the antibody together with protein A or G beads. And after you've chosen your antibody, and now I would like to tell you a little bit more about nanobodies and why nanobodies are so special for IP and so well suited. So I'm not sure if you're familiar with what a nanobody is. I think you all know the traditional antibodies that consist of a heavy and light chain, and alpacas and other cameloids also have, besides the traditional antibody, another type of antibody which only consists of a heavy chain and is called a heavy chain antibody. And the binding domain shown here in green of this heavy chain antibody is a nanobody. The nanobody is only 15 kilodalton in size, and it only consists of a single chain. So you don't have the heavy and light chain. And this makes it very suitable for IP. I will show you on the next slide why it's great that you don't have heavy and light chains. And when the nanobody is conjugated to a bead, we call this a GFP trap. And under the brand Chromotech, we have developed many different nanotraps. So we call them nanotraps, not only against GFP, but also against mCherry or RFP, or against smaller peptide tags like V5, for example. And in the next slide, I would like to show you how the IP looks different when you have a nanobody in your IP reaction or an antibody. So first of all, we already discussed that you need to immobilize an antibody on the beads with protein on protein A or G beads. So you need to have a pre-incubation step to get the antibody bound to the beads. With the nanobody and the nanotrap, these reagents are ready to use, so you don't need to do the conjugation step. You really can just take the beads as they are, briefly wash them, and then start your immunoprecipitation reaction. When you now look on an SDS page, so how it looks like after the IP, we just look at the bound fraction in the middle. For an antibody, after the elution with SDS sample buffer, not only the protein of interest, in our case here, the GFP protein is eluted, but also the heavy and light antibody chains are released from the beads. And these chains show up in SDS page. And you see here in our example that the signal intensity of the heavy and light chain in some cases can even be brighter than the actual protein you're looking at. And it can make it very hard to decipher whether the bands you see on SDS page are your protein of interest or maybe belong to the antibody tool that you've used. Now, when we look more on the left side with our nanobody, first of all, the nanobody is really small, so it doesn't really show up on SDS page. And second, because of the covalent interaction of the nanobody to the beads, the nanobody remains bound to the beads even after elution with SDS sample buffer, and you only get one single band in SDS page. And this single band is in your protein of interest. And the third difference between an antibody and a nanobody IP is that antibodies usually bind their targets very tightly. They have a very high affinity, which leads to an effective pull down. So when you look at the Western blot at the bottom, you see that there's nearly no protein left in the flow through. So it's completely depleted, completely enriched on the beads. And when you compare it with an antibody IP, you see that there is an enrichment of the protein of interest, but also there's still some protein left in the flow through. And then nanobody is more efficient here. So now you may ask the question, when should I use a nanobody or an antibody for IP? And there's really it's also about personal preference, but our rule of thumb is that nanobodies are very well suited for tag proteins. For example, if you have a protein that carries a GFP tag or an M-cherry tag or a smaller peptide tag like MYC or V5 or the spot tag. And then we recommend that you use the nanotrap, so the nanobody for the IP, and use a conventional antibody for Western blot detection. For endogenous proteins, so when you want to use an untagged target, then usually an antibody works much better because there are more antibodies available on the market. But think about if it's possible that you can combine two different antibodies. So use one antibody for the immunoprecipitation reaction, for example, the monoclonal version of the antibody, and use a different antibody for Western blot detection, for example, the polyclonal version. If you use the same antibody for the IP and also for the Western blot detection, any unspecific binding of the antibody that is observed during the IP possibly also then shows up during Western blot detection, and then you don't get a clean result after the analysis. So now I would like to go through all the different steps of an IP together with you and also how you can optimize the steps. The first step of an IP is after you've chosen your tool that you would want to use, the preparation of the cell lysate. So you crack your cells, and then you remove the cell debris and you end up with a soluble fraction in a tube. Then you add your antibody or nanobody tool conjugated to the beads to this cell lysate and incubate possibly for 1 or 2 hours and mixing. After this, you wash away unbound molecules, so you have only your enriched protein of interest bound to the beads. And then in the last step, you elute your protein of interest from the beads and from the antibody. And typical downstream analysis are SDS page and Western blots, but some customers also perform mass spec. Let's start with the first step of the IP with the preparation of the cell lysate. Again, the protein of interest is expressed in the cell and is released from the cell during this step. And you have then the protein of interest in the soluble fraction. And there are different methods and buffers which are optimized for different cell types and also for your protein of interest. So generally, it's recommended to start with a mild buffer and add some detergent to keep the protein happy in the solution. Depending on which cell type you use, also the cell material can differ a bit. So we recommend that for one IP reaction, you either use 500 microgram total protein or 10⁶ to 10⁷ mammalian cells. When you work with other cell materials like plant cells, for example, because of the low expression level, we usually recommend that you really use more in a gram range of plants. For example, like 10 grams of plant material are pretty normal, as I said, because the expression level in plants is usually very low. And we recommend that you always use a fresh cell lysate, so avoid freezing the cell lysate. I know that's not always possible, but usually the results are much better and the background is lower when you have fresh cell lysate, because during freezing also other cellular proteins unfold and aggregate, and this can increase your background. After you have completed the cell lysate, then you add your antibody tool to the supernatant. So the antibody binds to the protein of interest, and other cell components remain in the supernatant. And also here are some do's and don'ts. So we recommend that you perform the IP reaction for 1 hour and keep the beads in suspension, for example, by slow rotation. If you incubate longer, and if you have more like a mixing is too low or too high, then you always can increase the background. So avoid settling of the beads, but also avoid mechanical stress. The volume of an IP is usually 200 to 500 microliters. If you work in a 1.5 milliliter Eppendorf tube, then you can ensure proper mixing. With a lower or higher volume, usually the mixing is not done so well in this tube, with this tube size. And using doing the IP at 4 degrees and using a mild buffer is recommended to keep everything happy in solution and also to avoid unfolding of proteins. Next, in the washing step, then you remove unbound cell lysate components. They are washed away and the protein of interest remains bound to the beads. And the rule of thumb is that usually you should perform the IP. So when you do the binding, that you have a very mild buffer, but for the washing step, you use a harsher buffer. So for example, with a higher detergent amount or higher salt concentration, to remove all the proteins and cellular components that bind unspecifically to your antibody or to the beads. And washing for a few minutes and 3 to 5 repeats, and with roughly around 1 milliliter is recommended. If you wash too long, or if you use too many repeats or use a high volume, then you can facilitate that not only unbound molecules are washed away, but also that your protein of interest is released from the antibody. And that's something for sure you would like to avoid. The last step is the elution of your protein of interest from the antibody and from the beads. And there the protein of interest is released. There are different options how you can elute. The most common one is denaturing elution with SDS sample buffer. And during this elution method, the antibody and the protein of interest are both denatured, and the protein of interest is released. Another also common option is to use acidic elution, for example, 200 millimolar glycine buffer. During this elution method, the interaction between the antibody and the protein of interest is disrupted. And depending on the nature of the protein of interest, it either also unfolds under these conditions, or it maybe still stay in the right structure and the native state, and you could get a native protein of interest during this elution method. The last method is proteolytic elution, for example, using a TEF protease. So in that case, you have to already introduce a cleavage site between your protein of interest and a tag when you start the cloning. So way before you do the immunoprecipitation, and I already mentioned then the protease that you can add on beads. So you can directly incubate the beads and the antibody with the protease. Then the tag is cleaved off, and the protein of interest you can find then in the eluate. And usually under these conditions, the protein of interest is in the native state. And after you have completed the elution, then you can do the downstream analysis to see if your IP worked and how pure it is. You can do SDS page and Western blot, but also some customers do mass spec. On this slide, I would like to show you how your analysis looks like on SDS page and Western blot. So on the left side, we have SDS page, and on the right side, we have Western blot. And in our example here, we have a GFP tag protein of interest that is probed on Western blot with an anti-GFP antibody. And to make it more simple, we used here in our example a GFP trap, so a nanobody for immunoprecipitation. The first fraction that you analyze is the input fraction, which contains a total cell lysate. You will see on SDS page many proteins. And when you probe this lane on Western blot, you should get maybe a tiny band in the input fraction of your protein of interest. So in some cases, you cannot see the protein of interest in the input fraction in case it has a very low abundance. But usually when you use a polyclonal antibody in Western blot, then you should be able to see at least a tiny band. The next fraction you should analyze is the flow through, which is the supernatant after you have incubated your cell lysate with the antibody or nanobody beads. You won't see usually a difference in SDS page. So there is a depletion in the flow through, but often you can't see it. So in many cases, the input and flow through fraction in SDS page look completely similar. But on Western blot, you should not see any protein of interest left in the flow through if your IP was efficient. And the last fraction is the bound fraction that you should analyze. On SDS page, if you're lucky, so if you have a high abundant protein, you can already see one band, which corresponds then to your GFP tag protein of interest. And when you look on your Western blot, then you should see a single band of the protein of interest, which is enriched, so which should have a higher signal intensity compared to the input fraction. So this is the normal analysis that you should do on SDS page and Western blot, but controls are also essential for immunoprecipitation. And one very typical control is to see to do a positive control and see if your whole IP setup works. So during the positive control, you would like to see the enrichment of the protein of interest. And when you work with tags, then you can use the tag only, for example, GFP. And when you work with endogenous proteins, then it's more difficult to find the right positive control. But what you could do is use purified or recombinant protein of interest and spike it either into a MOX cell lysate, or you just use it in buffers so that you have no cells present in your positive control. And when we look on SDS page and Western blot, because this is a positive control, what we would expect is that we have our protein of interest in the input, nothing in the flow through, and an enrichment, so a stronger band in the bound fraction. When the positive control is successful, then you know that your whole IP setup works, so that you've chosen the right buffers, for example, and also that the incubation temperature and the incubation time that you have chosen is right for your assay. More important than positive controls are usually negative controls, and the first negative control I would like to show you here is that you want to check that you have no unspecific binding of any cellular proteins or your protein of interest without the when no tag is present or when you don't have your protein of interest present. So when you work with tag proteins, then we recommend that you use a cell lysate that contains your protein of interest but does not contain the tag. And when you work with endogenous proteins, you could do a genetic knockdown or knockout. And when we look on our Western blot here, in our example, we have there's no GFP present. And of course, then when you probe your membrane with the GFP antibody, you are not supposed to see anything. So this is also good control about the quality of your Western blot antibody. You could also, in that case, use an antibody against the protein of interest. And then in our example here, you're supposed to see it in the input and in the flow through, but not in the bound fraction. Another important negative control is that you want to check that you don't have any unspecific binding to the bead matrix itself. So you can use, when you work with a nanotrap with the tag protein, you can use blocked beads. And when you work with an antibody, then you can check how the protein A or G beads, what they bind in the absence of the antibody. This is called usually pre-clearing, but you can also use an antibody isotype control. So an antibody that has the same isotype as your IP antibody, but detects a different target. And then also together with protein A and G beads, this is often called a pre-clearing step. And when you look at the Western blot, then you are supposed to see your protein of interest only in the input and in the flow through, but not in the bound fraction. When it comes to co-IP, then controls look a little bit different. So again, just as a reminder, what is co-IP? Co-IP is used to enrich and identify the interaction partners to a bait protein. So in that case here, we use a GFP bait protein, and the interaction partner we want to analyze is here shown in red, the prey protein. And during a co-IP, the GFP bait protein in our example is directly precipitated by a nanobody or by an antibody, and the prey protein is only indirectly precipitated by GFP bait. And bound proteins, both the bait and the prey protein are usually analyzed by Western blot or mass spec. So when you do a co-IP, you often don't look at the SCS page because it doesn't tell you so much. And in our example here, I would like to show you how the controls look like for co-IP. And this is really a very, very simple example. So in many cases, customers use a more like extended assay where they run more controls. So this is the absolute minimum I would recommend that you do when you run a co-IP. And here, usually also you do then 2 Western blots. So we show it here as one example, but you run one Western blot where you incubate the membrane with your antibody against the bait protein. In our case, again, we use an anti-GFP antibody to detect GFP bait. And then you also use a second Western blot to detect the prey protein with an anti-prey antibody. And when you start the co-IP, again, you would possibly start with a positive control. So you will check for the precipitation of the GFP bait protein. In the input fraction, you see that the GFP bait is present, but no prey is present. And when you analyze on Western blot, you only get a band for GFP bait. And also for the IP, when you look at the Western blot, you're supposed to pull down and enrich your protein of interest, so GFP bait, on the beads, and you're supposed to get a band then also in the input pull down fraction. The second control is a negative control. There you check that you don't precipitate your prey protein if no GFP bait is present. So in the input fraction, you only have the prey protein, which you should detect also then on Western blot in the input fraction. But when you look at the IP fraction, you are not supposed to get any band for GFP bait because it's not present, but also not for prey, because the prey protein should not be pulled down if no bait is around. And the last case here is then your actual co-IP experiment. So you have both in your cell lysate the GFP bait and prey present, and you should detect both bands, also both proteins on Western blot and the input fraction. And when you compare it then with the IP fraction, also there both proteins should show up. And if as I said in our very simple example here, if the Western blot looks like this, you can be sure that the prey protein and the bait protein really interact, because you could exclude that the prey protein is unspecifically precipitated by either the matrix or the antibody. Okay, now I would like to come to the last part, to the troubleshooting part, typical trouble that may occur during your IP. I worked on 4 different examples, which are common, but for sure there are more. And in the example here, we will again stick to our GFP tag protein of interest, which is precipitated by GFP trap. And we will analyze the Western blot with an anti-GFP antibody. So let's start with the first one, which is really very common. I think that's one of the most common cases, that you have high background in your bound fraction. So the SDS page will show you in the bound fraction, when you look on the left side, that you have more than one band that you actually want to get in your IP. So you get additional bands in the bound fraction, and in worst case, they not only show up on SDS page, but also on Western blot. And the reasons for high background can be that your cell lysate has a high concentration, so that, for example, you use too many cells, or you didn't dilute your cells after cell lysis, or that the cellular proteins or also the protein of interest are degraded or aggregated. So when they're degraded, when your protein of interest is degraded, you usually get a lot of smaller bands below the molecular weight that you expect. And troubleshooting, so what can you do to resolve this issue? You could optimize your cell lysis. So for example, test different methods for the different cell types, and also try to find the right amount of cells and or of cell material that you need to use for an IP. For the binding step, when you have higher background, we recommend that you try to shorten the binding, so incubate at 4 degrees and incubate for maximum 1 hour. The longer you incubate, usually only the background will increase. So in many cases, incubating longer does not lead to a more efficient IP, but only to higher background. And when you see a lot of unspecific binding, you can also perform pre-clearing, for example, with the protein A or G beads, or in our case here with blocked beads. And it can help to add protease inhibitors, especially if you see that a lot of unspecific binding occurs like, with a smaller molecular weight than the actual protein of interest you're looking at. And when you have high background, what also helps a lot is to increase your washing conditions, so to use more stringent washing, for example, increase the salt or detergent concentration in your wash buffer. And what works like magic is that during the last washing step, you switch to a new tube. Because in many cases, unspecific binding that you observe does not only happen with the tool that you use, so with your antibody and the beads, but also your tube walls can be involved. So the plastic consumables that you use can also increase the background of your IP. Now, also a second case I would like to discuss is a very common case that there's no GFP protein, like no protein of interest is found in the bound fraction. So the protein of interest remains in the flow through. When you look at at SDS page, you will not see any band in the bound fraction. And when you look at the Western blot, you have your protein of interest in the input and in the flow through, but nothing in the bound fraction. And there are different reasons, different explanations. The GFP protein of interest could be insoluble or unfolded, aggregated or degraded. This already usually happens during the cell lysis, so not during the IP. Another possible explanation is that you have insufficient mixing, so that you didn't mix enough to really have exposure of the antibody to the protein of interest. Also, in some cases, you didn't use enough bead slurry, so enough of your tool, so which means that you exceed the binding capacity of the antibody or of the nanobody tool that you use, and if you use more, then you should get a band in the bound fraction. And a more rare case could be that your protein of interest is too large to enter the pore matrix. So this is usually more in the high, like in the megadalton range, I would say, or more you experience this if you have a large protein or also a protein complex that is formed that is more above 200 kilodalton or 500 kilodalton. So this doesn't happen with smaller proteins. And how can you resolve that issues? What can you do when your protein of interest is unfolded or aggregated? Then it's helpful to test different protein expression methods and different cell lysis methods. So that's you usually can't do anything during the IP. You have to resolve this issue before you actually start the immunoprecipitation. And for the binding reaction, so in case the problem occurs during binding, we recommend to mix properly, so to mix by end-over-end rotation. Mixing in a normal mixer usually is not, doesn't really help because the beads easily settle and then you don't really have a mixture between your supernatant and the beads. And we also recommend that you always use 25 microliters of slurry for one IP reaction to get this efficient pull down. And it may be a good idea to test different bead matrices, as I mentioned, compare porous beads versus solid beads in case you work with large proteins or protein complexes. The third case I would like to discuss with you is that there is no protein of interest in your assays, so you cannot detect it. When you look at SDS page, it looks similar to the previous case. So I would like to go one slide back. So when you compare here, the SPS page looks similar. You have you have bands in the input and flow through, but nothing in the bound fraction. But when you go on to the third example here, you see SDS page looks similar, but the Western blot looks different. This is one reason why analysis on Western blot is usually really helpful to diagnose what's going on with your IP and to make really statements about trustful statements about your experiments and this and the results. So coming back to the third example here, no protein of interest can be detected. So you cannot find the protein of interest either in the input or bound or flow through fraction. So your Western blot is completely empty. And what one possible explanation is that your protein of interest concentration and amount is too low, so that you may have a very low expression level. And the other explanation could be that the protein of interest is there, but it's insoluble. So after you did the cell lysis, your protein of interest is not found in the soluble fraction, but in the cell debris pellet, in this unsoluble fraction. And what you can do is to test different protein expression and cell lysis methods, and also maybe analyze on SDS page and Western blot the cell debris fraction to see if there if you can find your protein of interest there. And if your concentration is too low of the protein of interest, it usually helps to use more cells or a higher concentrated cell lysate. And what could also happen is that your Western blot antibody doesn't work so well. So therefore, a positive control is very helpful, and also testing different Western blot antibodies to find a really good one that detects your protein of interest well. And now coming to the last case is that you have a low amount of protein of interest after the elution with SDS sample buffer. So you may see it in SDS page, you may get a band, but the band is really tiny. And when you look at the Western blot, then you will see that although there's a band in the input fraction, the band in the bound fraction is much smaller compared to the input. So it looks like there's no enrichment of your protein of interest during the IP. But if there was no enrichment at all, you would also see the protein of interest in the flow through. So here, clearly something is wrong with the elution. So I would diagnose from this plot that the majority of the protein of interest is still bound to the beads because of insufficient elution. And this happens actually quite often, more often than you would think. To ensure proper elution, we recommend that you use usually at least 50 microliters of SDS sample buffer to ensure proper mixing of the SDS sample buffer with the beads. And also mix the beads properly with the pipette. So when you add the SDS sample buffer, mix properly with the pipette before and after boiling. And boil for 5 minutes, 5 to 10 minutes at 95 degrees. So standard boiling conditions when you do an SDS page should work well. So these were the 4 troubleshooting cases I wanted to discuss. And now I would like to wrap up. So the take-home messages of today's webinar are choose the appropriate nanobody and antibody tool for your IP. So not every antibody or nanobody is suitable for IP, and you have to find the right tools to do a successful IP. And I think you could also see from the troubleshooting cases that sample preparation affects your IP results. So it's not only about doing the IP, but also about the way how you prepare your sample. This can influence your results. And when your IP doesn't work well, think about that you may have to optimize each step of the IP to get optimal results. So usually a lot of things can be done during the binding step and during the washing step, but sometimes you also have to go back to the cell lysis and protein expression step. And not only like controls are not only key for meaningful results. We have seen already that Western blots really can make a difference when you analyze your IP reactions. Controls are also key for troubleshooting. So running all the controls can help you a lot to identify what's going wrong with your IP. And if you now would like to start an IP, or if you already did some IPs but would like to test maybe also other, maybe want to test the nanobodies for IP, under the Chromotech brand, we offer immunoprecipitation reagents for different fluorescent protein tags, for example, GFP, M-neon green, also M-cherry or halo and SNAP. But we also have nanotraps against other small peptide tags like the flag tag, MUG tag, spot tag, or V5 tag. And when you would like to run an IP with an antibody under the protein tag brand, we have more than 4,000 antibodies validated for endogenous targets, and more than 3,800 antibodies are validated for IP. And we also offer antibodies against 13,000 targets for Western blotting. So maybe one idea to use, for example, a Chromotech resin for IP and use a protein tag antibody then for the detection on Western blot. And on one of the last slides, I would like to show you that not only we as a company are successful, but also our customers are. And customers have cited already in more than 130,000 publications with protein tag products. One of the most recent publications in EMBO reports was a paper about myosin 6. And in this paper, they also used the Chromotech GFP trap for immunoprecipitation. And we are also very proud that Protein Tech won the Researcher's Choice Award from SciDapp 2020, which demonstrates that our customers really like our products, which we very much appreciate. If you now would like to do an IP as an attendee of today's webinar, I would like to offer that you get a free sample of the Chromotech nanotrap. So you can use the link that you see here. And I believe my colleague Lucy already posted this link also in the chat function, and we will provide it to you later also in our follow-up email. And we also offer a free 20 microliter sample of any protein tag antibody. Please just email us at events@ptglab.com to request it. And with this, I would like to thank everybody. If you have any questions that we can't answer now in the Q&A session, you're very welcome to send your questions to support@chromotech.com or events@ptglab.com, or you can also email me directly. You'll find my email address here on the left side.